So all mitotic cells in your body are in the cell cycle. And this cell cycle is made out of 4 phases. So first, the cell enters the G1 phase, the G1 phase of the cell cycle. And during the G1 phase, the cell is growing. And as it's growing, it creates new organelles. It's making more organelles. And also is making the endoplasmic reticulum larger. And it's also expressing lots of proteins and enzymes. For example, it's expressing proteins and enzymes that prepare the cell for the S phase. So now the cell enters the S phase. And once the cell enters the S phase, now the cell starts to replicate its DNA. There's DNA synthesis and replication, so now the cell inside the nucleus will replicate the DNA. And we did this using enzymes that we made during the G1 phase. But now at the end of the S phase, we've replicated our DNA. Now the cell enters the G2 phase. And during the G2 phase, the cell is also growing. It's also growing in size and making more organelles, larger endoplasmic reticulum, more proteins, essentially preparing the cell for mitosis. So now the cell enters mitosis, and when it enters mitosis, that's when we go through cell division. So those are the 4 phases of the cell cycle. So again, going into a little more detail, first, the cell is in the G1 phase. And remember, in the G1 phase, what's going on? This should be characterized by growth. The cell is growing. And as it's growing, it's literally getting larger, but it's also replicating its organelles, making new organelles, more mitochondria, more lysosomes, larger endoplasmic reticulum and Golgi. And also during the G1 phase, we're expressing lots of proteins. There's high protein expression during this phase, especially creating enzymes preparing the cell for the S phase. So now the cell enters the S phase. And once the cell enters the S phase, this should be characterized by DNA replication. Now, during the S phase is when we replicate our DNA. So now we replicate our DNA using the enzymes we made during the G1 phase. Now, once we've replicated our DNA, now the cell enters the G2 phase. And during the G2 phase, there's also more growth. The cell is growing, and it's also creating lots of organelles, lots of mitochondria, and lots of lysosomes, and a large endoplasmic reticulum, and a lot of proteins. And now, by the end of the G2 phase, notice what we've done. We've doubled the DNA, and we've doubled the amount of organelles we have. So now that we have all this extra DNA and these extra organelles, now the cell's ready for mitosis. So during the G2 phase, we're preparing the cell for mitosis. So now we enter mitosis. Now we enter the mitosis phase, and now the cell divides. And it's okay, it can divide, because remember, we had extra organelles and we had double the amount of DNA. So now we can divide, and when we divide, each cell has enough DNA and organelles. So now the cells have divided. And now once the cells have divided, now they start the cell cycle all over again. That's why it's a cycle. So now the cells enter the G1 phase. And remember, in the G1 phase, we're growing, we're making more organelles, and we're expressing proteins. So now we can enter the S phase. Now in the S phase, we use those proteins to replicate the DNA. So now the cells replicate their DNA. Now they enter the G2 phase, where they're growing even more, making more organelles, more enzymes, more proteins. And now that we have each individual cell has double the DNA, double the organelles, and double the proteins, now the cells again, now at the end of the G2 phase, now they're prepared for mitosis, and now they go through mitosis, and now they divide again. And they can divide because remember, they had plenty of organelles and lysosomes and proteins, so now they enter mitosis, and now they divide again. And now we do this cell cycle all over again. Now each cell will again now enter the G1 phase and go through the whole process over again. And the way I like to think about this is during the G1 phase, the cell is preparing for the S phase. During the G1 phase, it's creating proteins and enzymes preparing for the S phase. And during the G2 phase, the cell is preparing for mitosis. During the G2 phase, it's creating extra enzymes and organelles and proteins preparing for cell division and mitosis. And just a little bit of terminology, G1 to G2, this was referred to as interphase. This is normally the cell is hanging out in interphase. And again, different cells are different, and some cells hang out mostly in the G1 phase, but this is interphase. So there's interphase, and then when interphase is done, then the cell enters mitosis, where it actually goes through cell division. And this isn't true for all cells. This is only true for mitotic cells like hepatocytes and epithelial cells and the hematopoietic stem cells in your bone marrow. These cells are mitotic and they are dividing. They are going through the cell cycle, G1 to S to G2 to mitosis, then G1, S, G2 mitosis. So this is true for mitotic cells. But there are other cell types that are post-mitotic, like neurons and muscles. So these post-mitotic cells are stuck in the G0 phase. So they just enter this G0 phase and they just hang out in the G0 phase. They aren't dividing. They aren't constantly dividing and making new cells. They're just hanging out and surviving in this G0 phase. So this is true for neurons and muscles. So again, different cells have different parts of the cell cycle that they're in. And something important to realize is, how does the cell know to go from G1 to S phase? So this transition from G1 to S, this is referred to as a checkpoint. And it's important because how does the cell know to go from G1 to S? And once the cell goes from G1 to S, it's really committed towards mitosis and to divide. So we need very careful regulation. We need very careful regulation to determine whether the cell enters from G1 to S. So there's a checkpoint. So how does the cell know to go from G1 to S? Well, there are these really important proteins referred to as cyclin-dependent kinases. And these proteins are kinases. So what do? Well, once they're activated, once these cyclin-dependent kinases become activated, then they act as kinases. So we know kinases phosphorylate proteins. And specifically, these cyclin-dependent kinases, once they're activated, they phosphorylate transcription factors. And once these transcription factors become phosphorylated, now they're activated and they can act as transcription factors and they can express genes. And specifically, they express genes that help the cell go from G1 to S. So that's how the cell goes from G1 to S. And again, just simplifying it, the point is the cell has these cyclin-dependent kinases. And at all times, during all stages, at all times, these cyclin-dependent kinases are hanging out. However, once the cyclin-dependent kinases become activated, once they're activated, now they activate a cascade, so now the cell can go from G1 to S phase. However, what determines whether these cyclin-dependent kinases are activated? Because we know mostly they're just hanging out. However, once they get activated, then we go from G1 to S. So what determines when they get activated? Well, in the beginning, early on in the cell cycle, we start developing these cyclins. So these are cyclins. Remember, this guy is cyclin-dependent kinase. This is a cyclin-dependent kinase. However, these guys are cyclins. So early on in the G1 phase, we have these cyclins developing. But as we continue with the cell cycle, the cyclin concentration increases. And then as we continue more, as we go through the cell cycle even further, more cyclins increase. So as we're going through the cell cycle, these cyclins are increasing in concentration. And again, they increase, they keep on increasing. And then eventually, once we've gone through a certain period of time, at the end of G1 phase, we'll have a lot of cyclins hanging out. So what happens once we've gone through this period of time where as we've gone through this G1 phase, we've developed more and more cyclins? Well, eventually these cyclins will bind to this cyclin-dependent kinase. And remember, this green guy was a cyclin-dependent kinase. It was a kinase that was dependent on cyclins. So once we've developed a large concentration of these cyclins, then the cyclins start to bind to the cyclin-dependent kinase, and now the cyclin-dependent kinase is activated. Because remember, it was dependent on the cyclins. But once we went through the G1 phase, and now we have a lot of cyclins developed. As we continued, we've made more and more cyclins. And once we've made a lot of cyclins, they start to bind to the cyclin-dependent kinase. Now the cyclin-dependent kinase is activated, and now once it's activated, we explained that goes from G1 to S. Now it can remember what it did. It essentially it's a kinase, so it phosphorylated those transcription factors, which turned on genes to create proteins that allow the cell to go from G1 to S. But however, how does the cell know to go from G2 to mitosis? Because this is another important checkpoint, this so-called checkpoint. So again, the cell doesn't easily just want to go from G2 to mitosis. There's careful regulation to go from G2 to mitosis. There's a checkpoint that must be overcome. So how does the cell know to go from G2 to mitosis? Well, it's the same idea. We have a different class of these cyclin-dependent kinases. So again, this is a different class of these cyclin-dependent kinases. So again, these cyclin-dependent kinases are always hanging out. They're always in the cell just hanging out. However, they're not always activated. But once they become activated, then the cell can go from G2 to mitosis. So again, what determines whether they're activated or not? Well, it's the same idea. Maybe when we're early in G2, we have these cyclins increasing in concentration. But as we continue with the G2 phase, we have more cyclins increasing. And then as we continue even more, now we have a lot of, once we're at the end of G2, now we have a lot of cyclins that have developed, so we have a large concentration of cyclins. And remember, now once we have a large concentration of cyclins, they can bind to the cyclin-dependent kinase. Remember, this is a kinase that's dependent on these cyclins. So once we went through the end of G2 and we've developed a lot of cyclins, now they can bind to the cyclin-dependent kinase. Now the cyclin-dependent kinase is activated, and now we can go from G2 to mitosis. And remember, once we have a lot of cyclins, they bind to the cyclin-dependent kinase. Now it's activated. Now, once it's activated, it can act as a kinase. It can act as a kinase phosphorylating transcription factors. Now these transcription factors are activated. Now they can turn on genes, which create proteins that allow the cell to go from G2 to mitosis. So this is the cell cycle. There are these 4 phases of the cell cycle, and there's careful regulation. And really, it's actually a little more complex to go from G1 to S. There are other important things that the cell has to keep in mind. For example, it needs to know that its DNA is good. It doesn't have a lot of mutations and DNA damage. So that's important before we start replicating it and when we continue. So again, it's a little more complex than this, but the point is there are these 4 phases of the cell cycle, and there's regulation of what determines which phase we enter. And during different phases of the cell cycle, there are different processes occurring in the cell.